Hello everyone, welcome to Global Compliance Panel's live webinar on GMP expectation for products used in early phase IND studies. My name is Nathan and I'm going to be your host today. On behalf of the Global Compliance Panel team, I would like to thank you for being part of this event. Today's webinar will be presented by Stephen S. Carrara. Stephen S. Carrara, a PhD, is the founder and principal of GXP Biotechnology, LLC, a consulting firm that works in the areas covered by the GLP and GMP of drugs, biologics, and nutraceuticals. Steve has over 30 years of experience in supervising quality control laboratories, including an animal testing faculty, and in performing GLP and GMP audits of internal and external testing laboratories. Steve has participated in the development of drugs and biologicals through all phases of clinical research and final product production. We are honored to have such a distinguished person such as Dr. Carrara with us to present the webinar. Before we begin, I would like to inform you about the program outline for this training session. This webinar is for 90 minutes duration. First, Dr. Carrara will take you through today's webinar, highlighting the areas that would be covered, and he would then share with you his presentation. We would like to inform you that all participants, once part of the teleconference, have been placed on mute and will remain so until the question and answer begins towards the end of the webinar. Meanwhile, if you have any questions, please feel free to send them to me over the chat and we will address them one by one during the question and answer session. 10 minutes of time will be allocated for Q&A, during which your questions will be answered. And let me remind you, if for any reason you get logged out of this training session or teleconference, please follow the same procedure to join in again. Now that we are all ready to start, I request Dr. Carrara to take it from here. Dr. Carrara?
Okay, great. Let me get my pointer again and wherever I can find it. Okay. Hello. I am presenting this thing this morning on a little bit of a chilled state. It's a bit cool here in California. And anyway, before we get started, let me present you with the warning. The following is an excerpt of several regulations and guidance documents. And I'm going to present to you parts of it, what I kind of look upon as highlights of these guidance documents and regulations. But really, if this whole thing is important to you, you really should get hold of the original document and take a good look at it. I will show you the references to these documents, so you should be able to get them. They're all available at the FDA website. Now, we're going to talk about early phase studies. And for some people, early phase are phase 1, phase 2. And actually what we're going to be talking about are studies right at about, they're before phase 1 in some cases, and in other cases they're during phase 1, but not quite what you would think of as normal phase 1 studies. So there's a whole grouping of things, including things like the GLP studies, which are your non-clinical studies that you need to perform before you get into clinical studies. Now, among some people in the business anyway, there's I guess an unspoken ethical rule about how you should do in vitro studies, tissue culture, etc., and convince yourself in the in vitro studies that your drug is safe. And then if you think it's safe there, then you go into animals. You shouldn't expose animals to a thing where you don't even have any idea of its toxicity or anything else like that. And then after the animal studies indicate that the drug is safe, then you can go into humans in a clinical trial. And so we're going to talk about studies right at the borderline of animals to humans, and before you get into the so-called pivotal clinical trials and all this. Now, the first guidance document here, it talks about guidance. It's industry, investigators, and reviewers. And the investigators and reviewers, well, they're FDA people, FDA investigators, FDA reviewers. And they talk about exploratory IND studies. And it talks about what kind of things FDA considers as being exploratory studies in humans. And you really do need to kind of consider the performance of these studies. You know, this isn't the movies. You don't just think up a drug and go and stab a human being and stick it into them. You need to approach this in an organized manner. And particularly, you know, it's disastrous if you kill a person in an early stage clinical. So you do want to kind of go at this in a thoughtful manner, and that indicates that you know what you're doing. Now, for the purposes of these studies, this is what they consider exploratory studies. And it's studies that involve very limited human exposure, and it's limited in two ways. It's limited in terms of the dose you give to the humans, and it also is limited in the number of humans and the time of exposure. Now, one of the points here is that this study has no therapeutic or diagnostic intent. You're doing microdose studies, screening studies, things like this. And this guidance document only applies to very early studies. And we're looking at things, well, for instance, if you're trying to determine the mechanism of action of an experimental drug or a new drug, you may be, you may have looked, for instance, in tissue culture, you may have looked in animals, and you've decided that, well, okay, the mechanism of action goes in a certain particular way, and that there's a certain kind of interaction with a given enzyme, things like that. And what you do in these exploratory INDs is that you will, at this stage, take human material and expose human tissue, and in an intact human being usually, to your drug, and to verify if you're following the same mechanism of action that you saw in an animal. You can do a lot of your early pharmacokinetic studies using these techniques. For instance, you get a group of candidate drugs and you screen them to see if they're going to interact with a particular tissue target or a particular enzyme, and take a look at their pharmacodynamic effects and things like this, rate of absorption, all that. And you don't do this at a therapeutic dose. You do it at a low dose just to verify that the uptake occurs, in an expected fashion. And then one of the things that you really want to look at is biodistribution. This is getting to be more and more critical as we look at drugs, and particularly for genotoxic effects. You want to know, does it distribute itself into the reproductive organs, and does it really go to the tissues you expect it to go to, and things like that. And you can do this, again, without having to expose the human to a full therapeutic dose. And here's the thing, this is done a lot of times just for screening candidate drugs. and to basically to see if the drug that you have really is worth pursuing. Now, the content of these things, because they're very early INDs, and especially with a new chemical entity, these studies are going to be the first in human studies. And as a result, when you make your IND submissions, they're not going to look for previous human experience, and they're not going to look for a discussion for this. And they're much more flexible at this point in terms of the information on human exposure that you provide. And what you're doing here, it's a circumscribed group of studies, and basically what you're saying to FDA is that you cannot go further in your development without performing these studies and getting the results of these studies. Now, of course, those of you in the generics industries, usually this is not a problem because you already know what the innovator drug showed. So you have that data at hand. But for a new chemical entity, you're sort of feeling your way along as you do these studies. Now, you should articulate the rationale for why you're selecting this particular compound to study, why you need to do these trials, and at that point, you don't know a lot about this product, so you're not going to have a full development plan at hand. But you should be doing a risk assessment. And part of that risk assessment that would be important is any information you have about similar drugs out there. If you're, coming up with a member of a class of drugs, and that class of drugs is known to have certain toxic properties, or at very high doses, for instance, to have certain side effects, you would have to include that in the discussion. And the other thing is this thing. Your IND is going to be withdrawn. What you do here is you submit an IND to do these exploratory studies. And then after the exploratory study is done, you withdraw the IND. In other words, this IND is not going to go on to phase 2 and phase 3. And so you withdraw this IND, and then you submit a new one. And this new submission will go into your regular phase 1 studies and phase 2 and phase 3. And at that time, your regular IND would use doses, presumably the therapeutic doses. You're still going to have to do a dose escalation study and maybe a full toxicity assessment. But at that point, you will use this early study to guide the nature of the full IND that you submit later on. Now, there's two types of studies that FDA has been looking at. They have single and multiple dose studies. You do a subpharmacologic or pharmacological dose, but you have a limited number of subjects, and again, healthy volunteers or patients. Those of you in clinicals, there are certain circumstances under which you might expose a patient, even though you're not trying to do a treatment. You want to expose a patient to see if they can tolerate a dose at a certain level. This is especially important in the case of metabolic diseases or diseases that are known to affect the metabolism of the patient, because you want to know, a healthy human might be able to tolerate a certain dose, but people whose metabolism is affected by the disease may not metabolize the drug in the same way. And so you might have to use a microdose study early on just to see what will happen in these types of patients. And then your repeat dose, you know, you're looking for things like how rapidly does the drug build up in the patient, how long does it stay there, how quickly does it pass out of the patient, etc. And you can do this actually with repeated doses at a subpharmacological level. And, you know, notice here, it says duration of dosage should be limited. And they're saying, for instance, 7 days. And for escalating dose studies, you're designed to investigate a pharmacodynamic endpoint, not determine limits of tolerability. In other words, you know, at what point do you get too many side reactions, or do you start to see toxic effects? You're not going for that. You're just looking for a pharmacodynamic endpoint. For the purposes of the exploratory IND, they're telling you this is the following CMC information that they're expecting to see in your submission. And, you know, there's one statement right in the beginning: whether you believe the CMC, the chemistry, or the manufacturing presents any potential human risk. And again, you know, a lot of this is based upon your knowledge of the type of compound that you're dealing with and the type of manufacturing that you're going to have to do. For instance, there are some compounds that are perfectly safe to the patient, but where in the process of manufacturing that compound, you have to go through certain stages that may be risky in terms of the workers, or if the synthesis goes wrong, you will generate a toxic substance that can carry over into your product. So this would be a case of a human risk relative to manufacturing. Now, other, you know, preclinical studies that you've done and other studies that you are aware of where you can quote the information in an FDA submission, these can be used. And you should discuss them and just say, what are you going to do to monitor for these risks? In other words, sort of a risk management program here. And you present the candidate product, the active ingredient, and you can do it in a summary report. And the summary report should contain the following information. And they sort of give you a list of the description of the product and what kind of things they want to see, things like the therapeutic class, and dosage form, information related to the dosage form, description of the formulation, what excipients are you going to use? And remember, even at the early stages, you want excipients that are, well, they're so-called GRAS substances, G-R-A-S, generally recognized as safe. Or if it's, you know, part of a formulation that's approved or licensed in the U.S. For instance, you may be dealing with a completely new excipient as far as you're concerned, but this excipient may have been used in some other formulations, or for that matter, it might be recognized as a food or a food nutritional supplement. And, you know, you'd want to discuss this. The grade and quality of the excipients you're going to use. Are they USP, NF, ACS grade? Now, you know, the traditional thing was that excipients were supposed to be in the national formulary. So they would have to have been NF grade. But that has kind of broken down. The whole thing here is that you need to be able to prove that the excipient is safe and that it doesn't affect your active ingredient, things like that. Now notice components intended to appear in the product and those that may not appear but are used in manufacturing process. This is, well, there are things, for instance, like alcohol, good old ethanol that you use. You can use it in the manufacturing of certain biologics, for instance. but because of religious and other reasons, you don't want to have this in your final product. And so, you need to show that you use it and talk about how you're going to remove it. And name and address, method of preparation of the candidate product lots. I mean, you can't just make the thing and use it and without talking about or telling FDA how you're going to make it. And, let's face it, you have to make it if you're going to use it in the preclinical study. Now, remember this. If the manufacturing method you use here to make the material for the preclinical study differs significantly from the procedure that you use later on in your clinical trials, like to make the large lot for your phase 3 or something like that, you're going to need to explain why. Why the difference and how the difference has affected your system. Now, at this stage, you can choose a system, but make sure it's going to be pretty close to your actual method of manufacture. And then talk about, your packaging procedures and your container closure system, because, this affects the stability and the long-term shelf life of your product. For your active substance, you need to know how you made the thing. Now, remember, for those of you who know about the DMFs, the Drug Master Files, your active, you might be buying it, for instance, the API, and in which case a DMF should exist, a Drug Master File that you can refer to. If you don't have access to the DMF, then you're going to need to provide all this information. For parenteral type products, especially the sterilization process and the controls are very important. Now remember, when you talk about sterilization, you're also talking about the removal or reduction of pyrogenicity of the final product. For the biotech products, you want to know things like source material, the expression system, your harvesting methods, and the critical thing, especially for products that you will eventually send to Europe, is the inactivation of potential viral contaminants. And you need to know if your manufacturing method will reduce viral contaminants or if it can remove viral contaminants. For those of you in small molecule type drugs, okay, this usually is not so much of a problem because your manufacturing method will not carry viruses over into the final product, and especially if it's not parenteral. But for those of you who make parenteral products and that are derived from biotechnology material, the ability to remove or prevent virus contamination is very important. Your CMC information, you need a quantitative composition of the product, and in part of this is you need a brief description of adequate test methods. And they go through the usual thing: identity, strength, quality, purity, and potency, and the test results or a certificate of analysis. In others, you can summarize the test results into a C of A and turn that in. And again, for the candidate lots, and especially the ones that are used in toxicology studies, because if you later on find that your product contains a contaminant that was not present during the time of your toxicology studies, then you're going to have a problem on your hands. So you want to make sure that you've got a good description of the product at this stage. And again, for biotechnology products, especially those that came from mammalian cells or animals, you need to know about viral contaminants and I will point out, in case you have forgotten, not only are we worried about viral contaminants, but prions are a problem. The spongiform encephalopathy agents are considered to be the same as viral contaminants. Now, in many cases, what you do is you use material that is TSE or BSE free, or not expected to contain these agents. And that's usually the easiest way of taking care of that problem. You need stability studies on the product, especially the stuff that was used in toxicology studies. And how are you going to evaluate the stability during the clinical studies? There have been a couple of unfortunate incidents at the GLP level where products that were unstable and actually were inactive were used in toxicology studies, and then they were found to be safe. And then a active preparation that was not safe was used on a patient. And as a result, well, there has been at least one death that we know about. The ophthalmic products, parenteral dosage forms, you need sterility and pyrogenicity tests. And remember, ophthalmic products, you also need to have procedures showing how you remove potential things like particles and things that can irritate the eye. Now, there's two scenarios under which you can provide the CMC. In the first one, this is the one where you're using the same candidate product lot in your toxicology studies and in your clinical trials. Now, the toxicology studies that they're talking about here, these are the ones that you will do in animals prior to going into clinical trials on humans. Remember, you need to show it's safe in animals before you try it out in human beings. If you're going to use the same lot in both the animal tox studies and then you're going to go into clinical trials with that batch, material will be qualified for human use based on the earlier CMC information that you put out for the toxicology studies, and the results of the toxicology studies, in part at least, will be used to interpret the potential for harm to human beings. And you need an impurity profile to the extent possible. And let me warn you about one thing. When you do these studies, make sure you keep enough retention material around, because at later stages in the game, if new light is drawn to potential contaminants or things that might cause toxicity problems, you're going to need to go back and show that your toxicology studies were conducted on preparations that contain the material, and therefore, you don't feel it's a problem, or if there had been a problem, you would have seen it. Now, if it turns out that your toxicology studies were carried out on preparations that did not contain the new contaminant, and you're seeing it later on, you may need to go back and redo your toxicology studies to make sure that there is no human effect. The other parameters can be studied on an as-needed basis, and you need to know, your impurity profile and, recommendations that are given in agency guidance, like this document, for instance. In the second scenario, you're using another batch. It's not the same as the one that are used in the non-clinical toxicology studies. You're going to do human tox now using a different batch from the one that was used in the animal studies. And you need to demonstrate through analytical testing that your new batch is comparable to the previous batch. And they give you a list of suggested, they suggest that you use the following types of tests. Now, notice this. It includes, well, what they don't say is includes, but is not limited to. In other words, these are the kinds of tests you should consider, but there's other tests that you should use or you may need to use in order to prove that your new lot is the same or has the same characteristics as the old, or the one that was used in the non-clinical studies. Now, here are some examples of safety studies that are conducted at these early stages. Clinical studies, PK studies, these are done with microdoses. And usually these microdoses, now either you have a very good analytical method that is capable of detecting very small quantities or low concentrations of these drugs, or you may need to use, for instance, some sort of radioactive tagging, or, well, PET, positron emission drugs are also used, especially for imaging. Now, these things can be used. These are the so-called microdose studies. You could be as much as 100-fold below what you expect to use in your actual clinical efficacy studies. But at the low level that you use, you don't expect any toxic effects. And because of that, you're given a lot more leeway in setting up these types of studies. Your clinical trials to study pharmacologically relevant doses. This is, you're starting to look at your candidate products, and you're starting to look at your lowest doses that you will put into a human being. And at this point, you're trying to figure out, are you going to see any toxic effects? Are there any unusual side reactions and this sort of thing? Now, you're not going for maximally tolerated dose. This is one of the things you need to establish during a dose ranging study in your clinical trials. You want to know what that maximally tolerated dose is. And in fact, in some regulatory jurisdictions, you need to define your maximally tolerated dose. And you're not going to do that at this stage. You're just getting in there to know, the early doses where you start to see pharmacological effects. And again, the evaluation would still be less extensive than what you need for a traditional IND application. And then the other one is mechanism of action, MOAs, related to efficacy. You can study a mechanism of action without going to the maximum or the pharmacologically effective dose. You can go in a microdose and trace binding, metabolism of a drug, and all this without ever exposing your patient to the maximum dose. Now, we then get into the next one, which is a guidance document on estimating maximum safe starting doses. This is always a question when you're dealing with a new chemical entity. You've got a new drug at hand, and, how much of a dose can you give? Now, in the movies, they always just run in there with a syringe and stab the patient, and, it's always amazing that the patient doesn't just flip over and die when that happens. But, the question is, how much are you going to start with? And remember, it's not too good to injure patients, especially during your clinical trials, because this tends to put an end to your clinical trials, which tends to put an end to your product development efforts, and which tends to put an end to your company in some cases. So you want to start off with a dose that you know or have a lot of confidence that this will be a safe dose, and get this off the ground. Now, this guidance document is actually quite long, and I'm only giving you a very short bit of it. And you really do need, if you're dealing with a NCE, a new chemical entity, or a new type of product, you really do need to take a look at this guidance document. Excuse me. You're looking at an algorithm, the process, and also the vocabulary that you need to talk to FDA with on what is called the maximum recommended starting dose, the MRSD. This is what you use for your first in human clinical trials of new molecular entities. You have adult, healthy volunteers, and the idea here is to determine what this dose should be. And basically what you're doing, is that you're going to start off with the dose that you know can be tolerated by in your animals. And if your animals can tolerate it, you then try and translate that into a human dose, and then you build in a margin of error, a safety factor into this. Now, the human equivalent dose, that's what this HED is. You look at a dose in an animal, in the animal model that you're using, and then you translate this into a human equivalent dose, and you use that to study your early phase 1 clinical study volunteers. Now, let me tell you one thing. You can calculate this HED, and what most people do, and you'll find that it's recommended in the document itself, people will actually lower this HED by factors of 5 or 10, depending upon the expected toxicity of the product, just to make sure that they've got enough of a margin that they don't really have a problem with a patient. And the process is given in a flow chart in this this guidance document. And I'm not going to show you the flow chart. It's too complicated. And I guess what I'm saying is that you really do need to take a look at it for yourself. Now, the idea here is what you're doing is you're determining an MRSD for healthy adult subjects, and then it's what you're going to use when you're beginning your clinical investigation. Now, notice the guidance is not pertinent to endogenous hormones and proteins at physiological concentrations, or vaccines for that matter. Because, with these things, we know what those should be. You're trying to you know what the normal concentration is in a human being, and so that pretty much tells you what your starting doses should look like. And the same thing with a vaccine. There is no maximum dose that you have to worry about. You're looking for the antigenically effective dose. But the process is primarily for drug products where you're going to have systemic exposure. In other words, like an oral dosage form or an IV form where you're going to put it in, and the drug will travel to all parts of the body before settling down. And like it says, it doesn't address dose escalation or maximum allowable doses. Overview, this Appendix E has the recommended process and the flow chart. Major element, you're looking for the so-called no observed adverse effects levels, NOELs, in tested animal species. Then you convert that, the no observed effective adverse effect level, into a human dose, an equivalent human dose. And there's a big question here, because when you do your non-clinical studies, these will be GLP studies conducted in animals. When you do these studies, there's always a question of, are you using the most appropriate animal species? In other words, everybody uses rats and mice. Wow, big deal, But the trouble is that in a lot of cases, rats and mice are not the best models for human beings. And in some cases, dogs are much better. You have to go to a slightly bigger animal. Mice and rats are convenient for most laboratories. However, when you're starting to get into serious non-clinical studies, you really do need to take a look at things like using dogs, using, sheep. Actually, pigs are preferred. In a lot of ways, physiologically, pigs are closer to human beings than a lot of the other mammals. And so you need to choose this so-called tested animal species to determine your no adverse effect level. And then you convert that, into a human equivalent dose, and then you apply a safety factor. And if you want to be really careful about the whole thing, you use a 10-fold safety factor. In other words, you reduce the dose even further, so you can be pretty sure that you're not going to get an adverse effect. Now notice that this thing says no observed adverse effect. It doesn't have to be a straight toxicity effect. It could be some sort of a side reaction. But whatever it is, you don't want to hurt the human beings. Now, there's situations where you need to modify this algorithm. And, the algorithm is designed for systemically administered therapeutics. Now, if you're going to use topical, intranasal, intratissue, or compartmental administration routes, and things like depot formulations, there are other considerations that need to be looked at, and the guidance document does talk about what you can do. Now, at the beginning, after you've collected your non-clinical toxicity data, you convert this into the MRSD, and then you look at the NOEL, the NOAEL. You use it directly to calculate your MRSD. And other data can affect your choice, and again, appropriate species. One of the things you got to remember, for instance, rats and mice are actually pretty tough animals. And there are substances that are not toxic to rats and mice that are toxic to human beings. There's other things like, for instance, one of the reasons we use guinea pigs in a lot of studies is because humans and guinea pigs are the two mammals that do not synthesize vitamin C and need to be, take vitamin C in their diet. And so there are certain metabolic pathways that are not present in guinea pigs that mimic the same thing in humans. All of these considerations have to be made. You don't just go out there and say, oh, rats are cheap, let's use them. The other thing is you have to take this no adverse effect level for each species, convert this into the HED. Now, for most HEDs, you normalize your doses according to body surface area. And there are factors when scaling from humans to rats, guinea pigs, dogs, etc. These scaling factors are talked about in the guidance document. And remember, a lot of times when you start your first early clinical studies, for instance, you will scale your doses based on the body surface area of your patients, rather than you don't do dose per kg body weight until much later in your studies. And well, it's been found that body surface area versus dose is a much better correlation than the dose per kg body weight in a lot of cases. Now, however, there are cases, where you might use dose per kg body weight, etc., depending upon what's more appropriate in a given situation. Now, you're basing your data on what's available in each individual case, and this normalization to body surface area now, here's the thing, it should be done in one step by dividing the NOEL for each animal species by the conversion factor. Now, this BSA-CF, body surface area conversion factor, this is given in a table in the guidance document. And it's a unitless number, and it allows you to convert the NOEL on a milligram per square meter basis into a human equivalent dose. And like I've got down here at the bottom, for the rest of the material, look at the original document. The problem with this document is that it's very important for some of you, and it's not so important for others. So, this is why I've kind of given it short shrift here. Now, here is a more recent document. This is effective September 2008, which seems like a long time ago, but for some people this is brand new information. Basically, what happened here is that FDA has proposed, well, they didn't propose, they actually came out with a direct final rule, and it's in the GMPs right now. But basically what they're saying is that they are exempting phase 1 drugs from complying with the full GMP requirements. Now, there's a thing that you will see as we go through this, which is it's one of these typical FDA type things where they tell you you don't need to do something, but then they turn around and make you do it anyway. It's there, FDA operates with sort of this schizophrenic approach where on one hand they think that they're being a friend to industry by letting people get away with not doing certain things. And then on the other hand, there's regulatory requirements or legal requirements that say that FDA has to do certain things. And they're sort of caught between that and this desire to be nice to industry. And what you're going to see in this document is that there are some, very useful things where they've said that you don't need to do it. But as far as the rest of the GMPs are concerned, they still expect full compliance. Now, let me point out one thing to you, because there's sort of an attitude among some of the manufacturing people, especially some of you who are in new startup companies, that you don't need to follow any GMPs at phase 1. And you can do whatever the heck you want at phase 1. You don't need to really follow GMPs until phase 3. This is not true. The Food, Drug, and Cosmetic Act itself, not the regulations, the law, states that any product that goes into a human being, and notice it does not exempt phase 1 products. It says any product going into human beings must be made according to good manufacturing practices. Now, what this means is that FDA has had to reinterpret the GMPs specifically for phase 1 products. And so that's what we're going to be talking about here in this guidance document. Now notice this, it exempts phase 1 products, but then they come right back and tell you they will continue to exercise oversight through a review of the INDs. And so the INDs are getting to be more important here. And, you know, these early phase studies that we're talking about, these would be phase 1 clinical material. So you would follow this document for the GMPs that you should use. Now, it says several reasons. Well, you know, for instance, well, okay, let's go through this thing first. First, it says you should be exempted from complying with 210 and 211, which are the GMPs. Now, then it says even if you're exempted, you're still subject to statutory requirements that says that a drug is adulterated if it doesn't conform to CGMPs. So, you know, you've got this weird situation of FDA saying you don't need to follow the regulations in 210, 211, but the main statute says that you still need to follow GMPs. So now they're going to tell you what the GMPs are for this type of thing. They're saying they're going to use the IND regulations, excuse me, to sort of regulate this thing, and they're going to look upon, you know, all these sections of, now remember 312 covers INDs, 314 is the one that covers the NDAs. Now you're still subject to review under the IND regulations. Now they have specifically amended 210.2, 21 CFR 210.2, and they've inserted this thing, this paragraph C, saying that an investigational drug, you know, is subject to the statutory requirements. Production is exempt from compliance with regulations in 211 of this chapter. And, you know, 211 are the full GMPs. Now, there's a couple of exemptions here. Notice this. Once you use that drug in either a phase 2 or a phase 3 study, that drug has to be made according to full INDs. Now, you know, there are situations where you can get into a late phase 2 or into a phase 3 study, and questions can come up about the product, and you're going to say, whoa, I need to go back and repeat some studies that I did at phase 1 in order to check certain types of toxicology parameters, or look for certain side reactions that maybe we didn't look for in the early stage in the phase 1 studies. In which case you have a product that's being made for phase 3 or phase 2 studies, and you're going to take this product and go back and use it in a phase 1 study. These products have to be manufactured according to the full GMPs. You know, as soon as you go into phase 2 studies, you are liable to the full GMPs, not just to these modified GMPs. Now, this companion guidance, this is kind of crazy. It came out in November of 2007, but the actual effective date of implementation was July 15th of 2008, not November of 2007. But this companion guidance is the stuff that really tells you what the CGMPs are for phase 1 investigational drugs. And guidance applies as a part of CGMP. It applies the quality control principles to the manufacture of phase 1. And it goes on and tells you these things. Now, notice here, guidance does not apply to the following phase 1 investigational products. Human cell or tissue products that are regulated under paragraph 361 rather, of the Public Health Service Act. Now remember, these are biologics in this case. And these are the biologics that are basically they're transplantation products, very simple human cell products. By that I'm talking about, for instance, blood transfusions, where you're taking human cells from one person and either putting it back into that same person or into another person. These are the ones that are not heavily regulated in the first place, but this guidance does not apply to them. It does not apply to clinical trials for devices. Anything under the device approval or clearance procedures is not covered. IND products made for phase 2 and phase 3 clinical trials. So now let me warn you about one thing. I know some of you are going to start trying to sneak around this by saying, well, we'll make one big lot and it's not going to be for phase 1. Well, it's for phase 1, and then we'll carry it over into phase 2. If you're going to do that, you need to follow the full GMPs. You cannot manufacture under these rules for phase 1 product and then carry it over into phase 2, because as soon as you go into phase 2, then the manufacturing of the product needs to follow the full GMPs. Again, already approved products. This is where you have to go back to clinical trials. For instance, example here is given for a new indication. You have a product that you're making for one purpose, and excuse me again. The product then gets used for a new indication, and you're going to go back and do clinical trials so that you can market it for that indication. When you do that, you're going to have to go back and do some phase 1 clinicals again, especially if you're going to need to look at the mechanism of action and stuff like that. So when you do that, you have an already approved product that you're going to go back to phase 1 clinicals with. However, since it's already marketed, you should be following full GMPs. PET drugs, the positron emission topography drugs, they're subject to the Food and Drug Act, and there's a new PET CGMPs. And here's the catch. There's going to be a 21 CFR 212. Not 211, 212, when finalized. So far, these PET GMPs have not been finalized. So, but then when they are finalized, they will follow that GMP, and this guidance will not apply to them. The federal guidance, the general guidance just says that, well, okay, adherence at phase 1 occurs mostly through, well-defined written procedures. Okay, you're probably going to need to do that anyway. Adequately control equipment and manufacturing environment, accurately and consistently recorded data from manufacturing, including testing. So, this isn't much different from regular GMPs. And look at it this way. You're going to have to go to regular GMPs anyway. So you may as well start off in phase one with these well-written procedures, etc. Uh, you know, it just doesn't make sense unless you're planning to fail. This is one thing several consultants have pointed out to me, that this strategy of not doing something because, well, you don't want to spend the money because if the product fails, you would have wasted your money. Well, the catch there is that you're assuming that the product will fail. That's when you make money. That's when you save the money. If the product goes forward, then you're going to have to redo everything to bring it up to spec. So why not do it in the first place? That's how you minimize your cost. Your manufacturing environment, pretty much this doesn't change things, very greatly. You know, your risk, your hazards and risks from your manufacturing environment, particularly the susceptibility of a phase one drug to contamination or cross-contamination. Why are they talking about this? Well, the problem is that a lot of times phase one drugs are made in small quantities. So they're not made in a full manufacturing environment. They're made in like a development lab, or sometimes even just in a regular lab at a laboratory scale. Large scale for a laboratory, but small scale in terms of manufacturing. And because of that, they're made in an environment where other chemical substances, other products are present. And there's a greater chance for cross-contamination or for like a adventitious agent getting into the product in the case of a biologic. And so this is, for instance, the equipment. If you have, well, say 5 or 6 different postdocs making different products, working on other products, other vectors, things like that, then the chance that you're going to cross-contaminate within that lab is very high. And so this is one of the big points of concern here for your manufacturing environment, is how do you isolate your process, your product. Now, FDA comes out with this statement. We recommend the following steps. Now, remember, when you see that term, we recommend, they're not saying absolutely that you need to do it, but if you don't do this, you need to explain why you didn't do it and have a justifiable reason. Okay, if you can do that, then you don't need to follow the recommendation. But remember, if you don't follow the recommendation, you have to have a document someplace explaining why you did not follow the recommendation. Okay, comprehensive systematic evaluation of the manufacturing setting, product environment, equipment, process, personnel, material, and identify potential hazards. One of the big hazards is where you contaminate your employees. And, having worked in a biologics, well, in a blood environment during the days of the early AIDS epidemic, let me tell you, a lot of times you need to be protected from your product, not the product being protected from you. Appropriate actions prior to and during manufacturing to eliminate or mitigate potential hazards, to safeguard the quality of the phase one drug. Your manufacturing environment should have adequate work areas. If you're working in a small lab, especially university type lab or hospital lab, and you're trying to jam a lot of work into the same space, you've got a tremendous potential for problems in terms of cross-contamination. And the diversity of the products and their manufacturing operations, not all environments may be acceptable for manufacture. You know, this is the thing. You need to evaluate this, and again, be able to defend your reasoning as to why you thought it was appropriate to manufacture in a certain area. Under CGMP, if you have a contract with another party, and this is, you know, for a lot of you guys, at phase one, a lot of times, this is what you do. You go to a contractor to do your early phase one manufacturing. And, you know, this is perfectly fine. I mean, I work for a virtual company, for instance, where this was our routine procedure, was to go to a contractor to manufacture our product in all phases. But the thing is that the manufacturer needs to realize that they're responsible for meeting GMPs. And now remember this, you, the company that lets the contract, you're still responsible for them meeting CGMP requirements. And the quality control function stays with you. You still release the product. Your contractor cannot release the product, or should not. You can't delegate your QC responsibilities. And, you know, assess the contractor to ensure that they have the effective quality control functions. And when they say assess the contractor, this doesn't mean send a letter or a questionnaire. Go out there and look for yourself, and talk to people, make sure that they know what they're doing. CGMP must be in effect for each batch of drug used during phase one. You establish manufacturing controls based on identified hazards. And you know, this is the thing, even at phase one, you see, you should have been developing some experience, right? I mean, you made lots for your non-clinical studies, you've been making stuff for your experimental work and for early development. So by this time, you should have some idea of what your manufacturing controls need to be and what kind of hazards you're facing and all this sort of stuff. And the hazard, remember, always look at the hazard from 2 points of view. One is hazard to the patient, and two, it's hazard to the workers or your own company. You know, let me tell you, it is no fun if you manage to contaminate your whole manufacturing area. And especially if you manage to, well, create illness in your own workers. This has very bad effect on morale. So, you know, these things should have been worked out during development, early development phases. And make sure that you've got your manufacturing controls in place. Now, we're going to go into a list. These are considered to be applicable to manufacture phase one drugs and some specific manufacturing situations. First of all, personnel, what do you think? The usual GMP stuff, education, experience, and training, any combination thereof. Now, remember, it's combination thereof. There have been some cases of companies that have claimed that they have trained people, people who had no education and no experience. But by pure training, they have qualified these people. This has happened in some cases where they have hired upper level managers, for instance, directors, with no prior education or experience in the business. And then they've claimed that they've given them 6 months worth of training, usually undocumented, and that they're not qualified to be doing this work. Well, FDA has come in on several cases and said, no, this is not acceptable. So, you know, You have to be able to perform the assigned function. And appropriate experience to prepare phase 1 investigational drug and familiarity with QC principles and acceptable methods for complying with CGMP. This is the whole thing, because people can say things like, well, I've worked in a lab for 5 years and I know what I'm doing. Well, you don't. Uh, Especially in terms of QC principles and knowing the GMPs. Many people do not, including people who have been working in so-called laboratory environments for 10 years or so. All they've done is that they've done the same old sloppy work for 10 years. And going into a CGMP environment is quite different from that sort of situation. This is why a lot of times during employment interviews, people will be asked, have you worked in a GMP environment? What kind of GMP experience do you have? Your QC function, now notice this, manufacturer should establish a written plan that describes roles and responsibilities for QC functions. And they tell you what they expect to see in your written plan. And, examining your materials, your raw materials, your container closures, your in-process material, your packaging and labeling, and all of this to make sure that they're appropriate and meet defined standards. Some people, and I've worked in places where, for instance, we use conical centrifuge tubes with screw cap tops as our product containers. And that's perfectly fine at this early stage. But, you have to define these things and know that they're appropriate. Conical screw cap centrifuge tubes made out of polypropylene are perfectly acceptable in some cases. With other products, they're not. Product tends to stick to them, for instance. Things like that can happen. Who reviews and approves manufacturing procedures, testing procedures, and your acceptance criteria? Remember, it's a joint function between manufacturing and your QC people or your quality people. Responsibility for releasing or rejecting each batch of product based on review of completed manufacturing records. This is the thing. You can't just write it down in your lab notebook. You should have a batch record for each product, each lot of product that you manufacture. And, who releases or rejects it based on a cumulative review. In other words, a QC/QA function. And remember, it's QC that is responsible for releasing product all the time, not manufacturing. Manufacturing has a conflict of interest. They made the product, so of course it's acceptable. QC plays the role of the second pair of eyes, and they're the ones that have the final say on the release of product for use. Now, responsibility of who investigates unexpected results or errors. This is the OOS problem, the auto-specification test result problem. Unexpected results or errors that occur during manufacturing. Who investigates and who handles complaints that are received? If you go into an early clinical trial, you can still get complaints, usually from the clinical trial personnel, who will say things like, boy, your product really stinks, or the worst one being, we can't get your product out of the vial because your screw caps are so tight that we can't open them. Other things like, oh, your glass vials cracked because you went through a freezing and thawing cycle when you shipped it to us. Things like this, there's all kinds of complaints. And who handles your complaints? who initiates corrective actions if necessary. And, it says, although quality is responsibility of all personnel involved in manufacturing, we recommend you assign an individual or individuals to perform QC functions. In other words, somebody better be named. Doesn't have to be by a personal name, but it can be by position. And you say this person is responsible for performing the QC functions. Now, the minute you do this, of course, make sure that their training file shows that they've got the education, experience, and training to be responsible for the QC function. Especially, the cumulative review and release of the drug batches. You cannot just take a person with very little training, no experience or education, and then, turn them loose and say, okay, you have the release authority. However, now, in very limited circumstances, depending upon the size of the organization, all QC functions may be performed by the same individuals performing manufacturing. In the early stages, you could use your manufacturing people to perform the QC function. Usually you will have to name, they will have to be designated individuals, manufacturing individuals who are named as performing the QC function for you. And, it might be necessary. You have the same individual doing both manufacturing and QC, and including the release or rejection of the batch. And they still, however, strongly recommend that another person not involved with the manufacturing operation conduct an additional periodic review. Okay, I'm a consultant, and here's my commercial. I mean, I have been called in a couple of cases on a periodic basis to come in and review these internal batch records just to make sure, as a second pair of eyes, to verify that the QC function is actually being performed. And, things like, manufacturing, there are QC requirements during manufacturing. Were they done? Were they recorded? Etc., etc. Activities such as testing commonly performed by dedicated QC people. If it's done by manufacturing people in phase 1, you should have adequate controls. Excuse me. Again, things like segregation of testing, and you don't want to contaminate your testing or negatively affect test results. And, you don't want to have any of this going on. Your manufacturing facility should have adequate work areas. And again, you can't just squeeze these guys into any old space. You have to have the following equipment. Notice that this pretty much repeats the facilities requirements of the GMPs. And, these requirements are not significantly different from the regular GMPs. Appropriate equipment that won't contaminate your drug or react with, you got to have inert surfaces, in other words. Identify all equipment, have procedural controls, and, again, you want to make sure that you're not getting cross-contamination or mix-ups in your facility. You have problems with control of components, containers, and closures. You need written procedures on how you're handling your material. Remember, your incoming components containers and closures. FDA has identified roughly 25% of manufacturing or GMP problems are because of the acceptance of inappropriate containers, closures, components, etc. You should be able to identify and trace all materials. You know, this is the other thing, because you come out of phase 1 and go into phase 2 manufacturing, you should still be able to show that the materials that you're using are the same as, you know, material used in the previous phases. And you should have a record or logbook on the relevant information and this sort of thing. Manufacturer should establish acceptance criteria for some materials. All relevant attributes or acceptance criteria should be known at phase 1. Now remember, there are requirements these days for you to identify very early on what are known as the critical quality attributes of your product. And there's, well, critical process requirements that lead or generate the appropriate attribute. And you need to identify these things very early on. In fact, they should be identified during your development stage work. And you know, and so this would be done, you know, here. It should be before your phase 1. Your development work should have identified these acceptance criteria, the importance criteria, quality criteria that you use for acceptance, etc. Take a look at your C of A on each lot of each material to make sure. Now, here's the thing. In normal manufacturing, you need to not only look at the C of A, but also perform an identity test. FDA is more or less saying, well, you don't need to do the identity test at this stage, but you should examine the C of A nonetheless. You know, amazingly enough, the C of A's do change from time to time. And sometimes it's because your distributor switches to a new supplier and all this sort of stuff. So you have to have this documentation available. You have to have manufacturing records, follow written manufacturing processes and control procedures. And this is all the stuff you need to have in your record. And changes in procedures and processes. You will notice that these things, like your microbiological control and you know, follow the recommendations for use of aseptic technique, control of in-process material, and all that. These things are not that much different from regular GMP requirements. Laboratory testing, you know, they will say that these procedures do not need to be validated at phase 1. However, the attributes of the test procedures need to be known. Now, you see, test procedures should be scientifically sound, specific, sensitive, and accurate, suitable and reliable for the specified purpose. Now, these are exactly the same criteria as those required for a test method validation. And so you have to ask the question of, if we're using unvalidated methods, are we going to be able to validate them by phase 2? And the question really that comes up is that if the method is not validated, in other words, you can't prove that it's specific, sensitive, and accurate, what does this do to the rest of your phase 1 studies and you know, your earlier studies? To ensure reliability, you should calibrate your lab equipment. You know, verify. You know, this is one of the requirements of GMP, right? Before you use a piece of equipment, You verify that it's in good working conditions, instruments when the sample is analyzed. You perform a system suitability, or you perform a maintenance check to verify that your equipment is working properly. Other laboratory things now. Notice this, retention samples. You retain them for at least 2 years following clinical trial termination or withdrawal of the IND application. And the quantity adequate to perform additional testing at a later date. And this one says, for instance, twice the quantity necessary to conduct release testing, excluding pyrogenicity and sterility. And I have been trying to indicate to you, you may want more than that, if possible, because if things later on in your studies, say at phase 3, a question comes up about the presence of a contaminant in your product. You need to know if that contaminant was present during your phase 1 studies, especially your tox studies. And you're going to need to go back and get some of these retention samples to see if that substance was present. And if you're starting to run out of these retention samples, you're going to have a real problem. So make sure you keep enough material around so that you can do these additional studies. You should have a stability study, using representative samples of the phase 1 investigational drug. You need to be able to prove that your product was stable during the period that it was used in your clinical study. That's the minimum requirement. You don't need to go like for a full 3-year stability or anything else like this, as long as you can prove that it was stable over the period of your study. So, but you do need to have a stability study going. Labeling and distribution, they don't expect you to have a full warehousing operation at this stage. I mean, after all, you're doing only phase 1 studies. It's small material. So a lot of the GMPs that refer to phase 1, warehousing, distribution, and all this have been removed. However, you should still be doing these things. So how do you control your packaging, labeling, and distribution? And again, they recommend the use of these appropriate measures to achieve effective control. And, here's the thing. You don't want to mix up your placebo or with your active product. Worse yet, if you're doing blinded studies with multiple strengths and things like that, you need to be sure that you are administering the appropriate material to people. Distribution, transport of the phase 1 drug to the clinical investigators. And this is what you're doing. This is not commercial distribution. So the regulations that apply to commercial distribution do not apply. But you still need to be monitoring your distribution to the clinical investigators. You should have a distribution record. What if you have to recall? And this has happened during a phase 1 or early phase clinical study where you need to contact people and get your product back because you just discovered a problem with it, all of this. So you need to have these records. Here's your record keeping, including but not limited to the following things. And you must retain records for at least 2 years after the application is approved, the marketing application. In other words, the NDA is approved. And finally, I've got a little bit here, guidance for IND meetings. You need to look at this because, especially at this early stage, you're going to go into what is called a pre-IND meeting. At least this is the smart thing to do. A lot of companies don't go into pre-IND meetings. Usually at this stage, especially the smaller companies, for some reason, people are afraid of talking to FDA, and they don't want to have a meeting. The thing is that it's smart to go in and talk to FDA at this stage, just so that you don't get blindsided by problems later on, during your studies. You don't want to have to hold up your clinical studies because of an unforeseen or unexpected problem. So really, your pre-IND discussions with FDA are really useful, and this guidance document basically tells you what they expect you to do in terms of setting up for these meetings. And with that, I will conclude this thing. I'm sorry for all this background noise that has taken place. Thought I had my phone turned off. But anyway, are there any questions? We'll turn this back to Nathan, and he will field your questions and send them on to me.
Thank you so much, Dr. Kuwara, for the wonderful presentation, and also all the participants for cooperating with us. As Dr. Kuwara said, this is time for question and answer to begin, and we request all who have questions for our presenter to click on the raise hand option, which is a palm-like icon at the bottom of your participant panel, or else you can send me a question over the chat and I shall put it across to the presenter to answer it. Meanwhile, I'll request all the participants to share your feedback through the feedback form that is on your screen right now. The feedback form has just 6 questions, mostly multiple choice in nature, and it would not take more than 2 minutes of time to answer. The polling will remain open till the end of the session, and you can answer it even after the QA session is over. We would be grateful if you could stay a little longer and complete this feedback for us. So dear participants, we are open for your questions. Please feel free to go ahead accordingly.
I will note to you that if after this is all over, a question does occur to you, feel free to send that question to the Global Compliance Panel, to Nathan, and he will forward it to me. And so if a lot of times right after these things are over, you think you don't have any questions, but then a day later it occurs to you, oh, gee, what, I should have asked this question. At that point, feel free to send us your question by email, and we'll be glad to take care of it.
Thank you so much, Dr. Kuwara. So, dear participants, you can feel free to ask your question at this moment, or you can always email us your questions, and we will put it across to Dr. Kuwara and get them answered as soon as possible. I don't see any questions coming up, Dr. Kuwara, so do you have any?
No, I think that's it.
Thank you so much. Dear participants, we are grateful to all of you for having taken part in this webinar. I would remind you, please share your feedback with us. The feedback form is open. And if you would like to get in touch with us, you can send your email to us at webinars@globalcompliancepanel.com. And if any of you feel that your team members, colleagues, friends might benefit from this webinar, we are happy to inform you that it will be available in the recorded format also and can be purchased from our website. I would like to take this opportunity to let you know about the upcoming webinars of Dr. Kuwara. Please feel free to get in touch with our customer support, and they will be more than happy to provide you further information in this regard. As you can see on your screen, you can reach us back at our customer support number. You can email us, so either way it will be fine. We welcome your suggestions and feedback or ideas on how we can improve our topics or the webinars. If you would like to suggest a topic or desire a customized corporate training whether it is online or on-site, we ensure that whatever is your training necessity, it will be our priority. So we look forward to having you with us again sometime soon, and for continued patronage. On behalf of our presenter, Dr. Kuara, and Global Compliance Panel team, I would like to say thank you for participating in this webinar, and we wish you a pleasant day ahead.
Okay, well, thank you, Nathan.
Thank you so much.